Hello, everyone. Welcome to this webinar organized by the American Society for Human Genetics. Thank you for joining today. My name is Alyssa Ortman, and I'm the Associate Director of Digital Programs here at the American Society of Human Genetics. Before we get started, I would like to take a moment to acquaint you with a few features of this web event technology. At any time, you may adjust your audio using any computer volume settings that you may have. You may also submit your questions in writing throughout the presentation by typing them in the Q&A box located on the menu bar. Also, we invite you to discuss the presentation with other audience members using the chat function. To open or close the chat, please use the button at the bottom of your screen in the menu bar. We are joined today by ASHG's President, Dr. Gail Jarvik. Dr. Jarvik is the Arno G. Mikulski Endowed Chair in Medicine, Professor of Medicine, and Professor of Genome Sciences at the University of Washington. At this time, I would like to turn the presentation over to Dr. Jarvik for opening remarks.
Thank you very much, Alyssa. And I'd really like to thank everyone for joining us today for this briefing. On behalf of the American Society of Human Genetics, or ASHG, I welcome all of you, those of you working in Congress, and those of you in genetics and genomics, and also those in the broader scientific and health community. If you're not familiar with ASHG, we're the largest organization for human geneticists. Our members are researchers, medical geneticists, genetic counselors, and others. Most of our approximately 8,000 members are in the United States, but we have a global reach. And the Society's vision is that people everywhere realize the benefits of genetic and genomic research. We know that human genetics and genomics, and science more broadly, not only leads to advances in human health and society, but drives the US economy. In the area of genetics and genomics, a report by the Battelle Group 10 years ago showed a considerable return to the American people for the US government's investment in the Human Genome Project. So today we are very excited to release a new report that takes a fresh look at the impact of genetics and genomics in the US economy and how progress in science is being deployed in both healthcare and the society. This report was commissioned by ASHG, and the work was conducted by Techonomy partners over the past several months. The report was very generously sponsored by Invitae and Regeneron, and ASHG is very grateful for their support in the development of this important report. I also appreciate that Dr. Bob Nussbaum, one of Invitae's leaders, is able to join us today. Today we'll have 3 speakers, and I will introduce all 3 before we get started. And at the end of the 3 presentations, we'll have time for questions. First, we'll hear from Dr. Eric Green, the director of the Human Genome Research Institute at the US National Institute of Health. Dr. Green is the 3rd NHGRI director and was appointed to his position by Dr. Francis Collins in 2009. He has played an instrumental leadership role in developing many high-profile efforts relevant to genomics, such as the Undiagnosed Disease Network and the US Precision Medicine Initiative, the NIH All of Us Research Program, and others. Dr. Green will speak to us about the promise and progress in genomics research. Thank you, Dr. Green, for joining us. The next speaker is Dr. Bob Nussbaum, who has served as Invitae's Chief Medical Officer since August of 2015. Prior to joining Invitae, he was Chief of the Division of Genomic Medicine at UCSF Health and Chief of Genomic Disease Research at NHGRI. He is a member of the Institute of Medicine and a fellow of the American Academy of Arts and Sciences. He is also a former president of ASHG. And Dr. Nussbaum will talk about some of the ways in which genetics is being used to advance health and growth in industry. And then last, we'll hear from Mr. Simon Tripp, Principal and Senior Director of Techonomy Partners. He is a highly valued and sought after person for his expertise in research methods, analytical insight, strategic thinking, and a broad knowledge of diverse fields in science and technology. He's a frequent speaker at technology-based economic development conferences and talks about impact analysis. He was the lead for this new report, and he also led the report that Battelle developed 10 years ago that I mentioned earlier. He will give an overview of the main findings of the new report. And so with that, I'd like to invite Dr. Green to give his first presentation. Thank you.
Well, thank you, Gail, and thanks to ASHG for inviting me to speak in this briefing. I'm delighted to be here and excited to share with you basically an overarching context for the two speakers that are going to follow by telling you in very brief fashion the progress that has happened in human genetics and genomics research over the past several decades. Now, appreciating the fact that we're probably dealing with an audience of differing degrees of detailed knowledge of genetics and genomics and science in general, let me just remind you, DNA, of course, is the information molecule of all living systems. All of the DNA of an organism is a genome. The study of genomes is the field of genomics. DNA itself is a molecule that encodes all biological information, but does so with 4 building blocks. We actually abbreviate these 4 chemical building blocks, A, G, T, and C. The way they convey information is through the precise ordering of very long strings of these building blocks, the G's, A's, T's, and C's across stretches of DNA. The human genome, so the human blueprint, if you will, consists of about 3 billion letters, 3 billion of these G's, A's, T's, and C's. And in the late 1980s, the field of genomics was born, and part of the excitement around creating this new field was this idea that the time was right to tackle the ambitious goal of reading out, or in other words, ordering or sequencing all the 3 billion letters in the human genome. That was organized as part of this large international effort called the Human Genome Project, which the US played a major role. The Genome Project was conducted over 13 years. It ended about 18 years ago, accomplished all of its goals, including the signature goal of actually determining for the very first time the order of the 3 billion letters in human DNA in the human genome. Now, when the Genome Project ended 18 years ago, we returned to one of the initial rationales for why we were going to do the Human Genome Project. And the idea was relatively straightforward, was that we saw opportunities for improving the practice of medicine if we had more detailed knowledge about the human blueprint, about the human genome. And whether you looked at the popular press or the scientific press that came out immediately near the end of the Human Genome Project, the idea of putting the words genomic and medicine together came to the fore. And the idea of genomic medicine being the next goal for this field of genomics was really pretty obvious when the Genome Project ended. To be honest with you, it was sort of blurry. We didn't really totally know what genomic medicine was going to be all about, but we could think about the kinds of things we might want to do if we could operationalize genomic medicine. And by genomic medicine, it's basically using unique genomic information about each individual and tailoring their medical care for that. We know that each of us carries around with us a slightly different blueprint, a slightly different genome, slightly different spelling of those letters in our DNA. And if we had knowledge of that and could figure out how that knowledge could improve the way we manage patients medically, perhaps we could improve medicine overall for the overall and advance the overall health of humans. But of course, that was an aspirational goal. We had another journey to embark on, and that journey only began with the end of the Human Genome Project, with the new finish line now being realizing genomic medicine in some meaningful way. And as with the Genome Project, we knew there were going to be many, many steps in this journey, and we don't even know what all those steps were necessarily going to be when we started it 18 years ago. But what I will tell you is that the last 18 years have put us several steps closer, and in fact, starting to see reality of genomic medicine being implemented. Now, among those steps that we've been quite successful in over the last 18 years, I'll just briefly tell you, probably the biggest one is the first one, and it was a technical advance. Because while we were successful in sequencing the human genome for the very first time by 2003, it came at a price tag of about $1 billion. We needed to reduce that price tag to something that would be a reasonable clinical test, something that could be done routinely for all patients getting medical care, perhaps something like $1,000. In fact, we came up with this idea of the $1,000 genome being an aspirational goal for genomics. The good news is that we reached that goal several years ago, and now you can pretty much routinely sequence a human genome for under $1,000. That million-fold improvement in technologies for sequencing DNA really has changed everything, because basically it is now fairly routine to be able to sequence a human genome. You could do it for under $1,000. You could do it in a day or two. Now, that's just getting the order of all those billions of letters. It doesn't get you the full interpretation. That we're still working on and improving. Much progress to remain. But technically, we can actually get a sequence of a human genome and use it for patient care now routinely and inexpensively. But many hurdles still remain for fully operationalizing it, but this was a very first important technical step. In order to really understand a patient's genome, we need to understand how the human genome works. I'm just showing you one slide that really exemplifies 18 years of progress that continues to tease out all the incredibly complicated biological information encoded by those strings of millions of G's, A's, T's, and C's. We have all these genes. We have all these ways of determining how genes are used, when they're used, what those genes are doing, what those genes are making, so on and so forth. I would summarize this by telling you that we've learned a lot about how the human genome actually functions, but we have a lot more to learn. My institute at NIH has this as a major priority for the coming decade or two or three, fully determining all of the biological complexities embedded in all that sea of G's, A's, T's, and C's. The third area of progress has related to understanding how our genomes differ from one another. There are spelling differences. Each one of us carries on the order of 3 to 5 million spelling differences compared to any other person on this planet. And we are figuring out what are the most common spelling differences, which of those is functionally important, and which of those is medically important. Progress goes on. We've made a lot of progress, but we are increasingly beginning to fully appreciate how we vary, how our spellings of our genomes differ, and how to operationalize that information, both for understanding how the genome works and understanding how to use that information about individuals for tailoring their medical care. Among those spelling differences, a very small subset of them play a role in human disease. Some of those play a role in more rare diseases, which typically are caused by spelling differences that break a single gene, diseases like cystic fibrosis and Huntington's and spinal muscular atrophy and others. And we've made great progress. Just to tell you how much progress we've made, when the Genome Project began in 1990, there were only 61 rare diseases that we knew what was mutated in the human genome. And today that figure is well over 5,000 human rare diseases for which we know what mutations are doing to specific genes to cause that disease. Tremendous progress. But there's more progress to be made, particularly with more common diseases that are not caused by mutations or variants in single genes, but are caused by variants that occur in multiple different places in the genome, much more complicated circumstances with what is often a greater contribution of the environment and social factors and lifestyle and so forth. And so these more common disorders really represent areas of great attention in the research community and great focus for the coming decade as we get better and better at teasing out the genomic basis of rare diseases, but in particular of common diseases. And of course, understanding the genomic basis of disease gets us incredibly close to operationalizing that information as part of routine medical care. And here I think we could be very proud of the fact that one of the ways this is going to happen is by having genomics being done broadly across the entire biomedical research landscape, not just by one institute at NIH, for example, or one research community, but increasingly many different parts of NIH, many different parts of the entire enterprise of biomedical research. And we have seen that dissemination really pick up pace since the end of the Human Genome Project. You know, we went from the earliest knowledge of what the human genome was all about when the Genome Project ended to seeing the threads of genomics get woven into all aspects of biomedical research and increasingly into human genetics research and translational research as we start to think about how to operationalize it. And now so many different disciplines, including medical disciplines, are beginning to embrace the use of genomics and weaving the threads of DNA into their routine work, including the work of implementing genomic medicine. And what I can tell you is what's very gratifying as we enter this new decade is that the concept of genomic medicine is no longer a blurry one. It has come into focus. We have seen some of the earliest applications of genomic medicine. I think most of it still remains ahead of us. We've just seen the tip of the iceberg, if you will, or the tip of the spear. But just these early examples of genomic medicine being operationalized has brought to clarity what it's going to be like. And it really sets up a very exciting circumstance of what we can anticipate in the coming decade or two as more and more opportunities for using genomics to improve medical care come to the fore. Before I close, I should just tell you that none of this progress I've just described to you happens without deep engagement with the scientific community and involving a lot of strategic thinking and planning. And about every 8 to 10 years, our institute, the National Human Genome Research Institute at NIH, brings together the community, spends a couple years of intense strategic planning, and then produces a scientific document that provides, if you will, a roadmap for going forward. And we actually just completed our most recent one, and I show you a URL here that you could go get this document. We published it in the journal Nature in late October, and it really provides, if you will, a roadmap of what's going to happen at the forefront of genomics, especially as it applies to human genomics and genomic medicine implementation in the coming decade. It's going to be a very exciting time, and I hope I've provided you a very nice context for this, stopping just short of describing examples where genomics is actually being operationalized in medicine, because my good friend and colleague Bob Nussbaum is about to pick up the baton from me right now and is going to now tell you some examples of where genomic medicine is becoming a reality. And then we'll hear from Simon after that. So Bob, please take it from here.
Thank you, Eric, and thank you, Gail, for the very kind introduction. I would like to talk to you about this next area that Eric was just talking about, and that is, what's the impact of genomics on medicine? Genomic medicine is to use genomic information about individuals in their clinical care. That's how it's defined. And we are already using it in many, many ways. And I'm just going to give you a few little tastes as we go through the session. Comprehensive, inexpensive genetic testing to diagnose genetic disease or identify people with increased disease susceptibility, including cancer. And I'd say there's no area in medicine where genomics has had a larger impact than on oncology and on the treatment, the diagnosis and the treatment of patients with cancer. But also finding the genes and the changes responsible for an increasing number of rare disorders. Rare disorders are rare, but in the aggregate, they're responsible for many, many billions, if not $1 trillion worth of medical care impact around the country. Knowledge of the contributions of genetic changes underlying common diseases. I'll show you an example of that, such coronary artery disease or adult macular degeneration, where people in their retirement time lose their vision, or diabetes. And using knowledge of genomic basis for an individual's disease to design precision therapy specific to the individual's genomic makeup. And I'd like to show you some examples. First of all, let's talk about diagnostics and prevention. This is a graph of the lifetime risk of developing breast cancer, depending on whether you have certain changes in a set of genes that are called hereditary breast and ovarian cancer genes or hereditary breast cancer genes. The gray line at the bottom is the population risk. It's what all women face just without any particular genetic changes. And the colored graphs you see above are the lifetime risks when they carry a change in a single gene. At the very top, of course, is the BRCA1. Our ability to identify women who carry these changes gives us an opportunity to provide them with appropriate surveillance and, if necessary, interventions to prevent the development of cancer. And not only that, if they, for example, have hereditary breast and ovarian cancer mutations such as in BRCA1, not only is their breast cancer risk, there's also a markedly increased ovarian cancer risk, which they would not be aware of if it were not the fact that we could diagnose and detect this change in their gene. Here's another example. So, finding genes responsible for an increasing number of rare disorders. On the right is a graph of data that I pulled down from the online Mendelian Inheritance of Man. This is a NIH-supported, NHGRI-supported knowledge base of genetic disorders that has been going on for decades. And what this shows you is over the last 20 years, the number of new entries, either genes that are identified as associated with disease or diseases associated with particular genes, has just continued to rise from the end of the Genome Project, we're talking about 2,000 to 3,000, up to well over close to 14,000. And this is directly a result of the information available through genomics. Here's another example. This is a database, a publicly available database supported by the NIH, NHGRI again. It is a database in where labs such as mine, Invitae, and other commercial laboratories, and academic laboratories that are doing clinical diagnostics put variants that they find in patients who have particular disorders into the public domain, de-identified. You cannot, the patients are totally anonymized. There's no way to know which patient this is. But the importance here is, just as Eric said, there are lots and lots of changes in a person's DNA. That doesn't mean that they all cause disease. In fact, the vast majority do not. So when you find a change, you have to interpret it. And that's what clinical laboratories do. And one of the ways to accurately interpret change is to share data with other clinical laboratories who are seeing the same kinds of patients, so that they can ask each other quickly and easily, have you seen this same change before in this gene in a patient with hereditary colon cancer? Have you seen it? And how are you going to describe this change? Is it one that you're quite sure is benign, doesn't cause disease, or do you think it's one that's actually responsible for hereditary colon cancer? There are over 1.4 million variants now have been put into this ClinVar database, and it is an essential component, and it's an example of how public information through genomics research and genomics support directly aids in the care, the diagnosis, and the care of patients. Here's another example. This is a graph that shows a 10-year rate of, or the chance of getting a heart attack, essentially, over the 10 years following your current age. And it's broken down into two basic groups. Those who have either a favorable lifestyle, an intermediate lifestyle, or an unfavorable lifestyle. It's the ones that you're all aware of: smoking, obesity, physical inactivity, and an unhealthy diet all represent unfavorable lifestyle. Favorable lifestyle is the opposite of all those. But there are also basic genetic contributions to coronary disease, which are determined by analyzing hundreds of thousands of variants around the genome, and together using them to form a risk score based on your genetic makeup for developing coronary artery disease. And what's quite interesting here, if you look at this, is, of course, having an unfavorable lifestyle, which is the orange bar, always gives you a higher risk for coronary events than having a favorable lifestyle, which are the blue bars. But you can see that the genetic risk also plays a major role. So if you were a healthcare organization and you were going to follow the U.S. Preventive Services Task Force recommendation that intensive intervention for diet and exercise, which requires a lot of work, that sort of intervention to really make it work with people, who would you first target for that kind of intervention? It's clearly going to be the people that have a higher genetic risk score from the beginning. So this really allows you to find out who's at the greatest risk and for whom would an intervention be most successful. So a summary of that article, of the graph I just showed you from Kara et al. Inherited DNA variation and lifestyle factors contribute independently to susceptibility to coronary artery disease. Healthy lifestyle is associated with a similar reduction of risk across each bucket of genetic risk. So this is very important. If someone has a high genetic risk, you can tell them, by altering your lifestyle, you can mitigate and reduce the risk that your genes, for which you have no control over at all, you can reduce that risk substantially. So it gives people the power to intervene in their own health. Now, let's also talk about another aspect, which is the impact on medicine, which is therapeutics. Knowing the gene and the changes in a gene that cause a disease allows precision targeted therapy. Nowhere is this more obvious than in cancer, where the identification of specific changes in cancer tissues that either drive the growth of those cells or allow the cancer cell to spread. Those particular genetic changes can be targeted very specifically with medications or with antibodies that will then slow or stop the disease. So we are on the cusp now and are already seeing, through the use of genomics, the genomics of cancer, we are seeing a revolution in the development of how we treat cancer. We are no longer just simply trying to kill rapidly dividing cells with standard chemotherapy. We are now transitioning into a time where every cancer is precisely studied using genomics, and then a particular precision target is identified, and if available, drugs used for them. But another area outside of cancer that I just want to mention is a devastating disorder called spinal muscular atrophy. It's the most common fatal recessive disorder in children after cystic fibrosis. And cystic fibrosis, of course, because of good treatment, antibiotics, and development of better physical therapy and respiratory therapy, cystic fibrosis is no longer a fatal disorder in childhood. Spinal muscular atrophy is. It essentially is a genetic disorder that destroys the same sorts of nerves that are destroyed in polio. And it's due to mutations in a particular gene called Smn1. It shows you a picture of essentially a newborn child, a drawing of a newborn child who's very floppy, has no muscle tone because he has lost so much nerve control over his muscles. What causes spinal muscular atrophy? It's sort of a complicated story, but I think it's worth going through because it highlights the point I'm trying to make here. There are two genes. One's called Smn1 and the other's called Smn2. They sit side by side in the genome. There is a single nucleotide or base difference between Smn1 and Smn2, where there's a C in what's called exon 7 in the Smn1, you have a T in that same position, in that same exon. Now, what's an exon? Dr. Green mentioned how genes contain sequences of bases which code for amino acids, which then go to make the proteins that do a lot of the work in the cell. But those genes are divided up into little bits or words that are called exons. When strung together, they form a sentence, which is the information that gene is trying to convey. So Smn2 has a single base change. As a result of it, that sentence is not properly put together. The piece, the word encoded in exon 7 gets skipped, so it's no longer in the protein. And as a result, the normal SMN protein, which comes from Smn1, is no longer made or is made at very low levels. And instead, an abnormal protein, which doesn't work, is called SMN delta 7. In other words, it's missing this exon 7. So one base change results in a second gene, which is almost identical, but because of that one base change, it does not make a normal protein. Now, what happens in spinal muscular atrophy? You lose both copies of Smn1. They're defective. So all you're left with is Smn2, which, as I just described to you, doesn't work well because of that single base change. So there's been a specific therapy developed where a nucleic acid is brought in, which binds to that region on exon 7, only at the T, and causes the cell to no longer skip exon 7. As a result, it includes now exon 7, and a normal SMN protein is made off of this Smn2, which under ordinary circumstances would not work. This is a highly precise therapy, only possible because we know the base change and can therefore engineer exactly the right therapy to come in and overcome the effect. So what are the enablers of genomic medicine? Well, they should be obvious from what I've just said. The technology that allows millions of base pairs of DNA to be sequenced simultaneously, that's what's responsible for the cost of a genome going down 1 million-fold that Dr. Green mentioned. Publicly available data essential to clinical care, such as GenBank, Dr. Green showed you pictures of GenBank data, ClinVar, which I showed you, and many, many others. But third is a Supreme Court decision in 2013, AMP v. Myriad, that overturned patents on using natural gene sequences to carry out genetic diagnosis. Why is that important? There is a huge lag between clinical research and clinical implementation due to patenting prior to AMP v. Myriad. For example, the famous hereditary breast and ovarian cancer syndrome, which Angelina Jolie was so instrumental in publicizing, the patent was issued in 1994. In 1999, researchers from Dr. Jarvik's institution from University of Washington, Mary Claire King, and others showed that the BRCA1 gene could be disrupted by large rearrangements, pieces of DNA that are either missing or flipped. The clinical test that had been put in place by the company that had the patent could not detect those rearrangements. 3 years later, they finally were able to detect 5 of the common ones, but still missed 12% of those rearrangements. In 2006, finally, comprehensive testing, including all rearrangements, made the clinical test much more sensitive. So that was a 7-year delay caused by a patent thicket that was unnecessary for the implementation of BRCA testing. And in 2013, the overturning of that patent led to a marked drop in the cost of the test from over $5,000 per gene to $250 today for all the genes contributing to hereditary breast cancer, done all at the same time in a single comprehensive report. Very similar kind of thing was seen for a fatal arrhythmia disorder. There are a number of young athletes that, for example, have collapsed on the basketball or volleyball court that have had long QT syndrome. The relevant gene was patented in 1997. Commercialization took 7 years because the patent was not acted on, then it was sold to somebody else who didn't act on it, it was sold to somebody else. Finally, in 2006, a second lab was able to secure a license to offer testing. So that's a 9-year lag because of unnecessary patent protection. It took 7 years and many misdiagnoses before comprehensive testing was made available. The sequence of all the genes in the human genome is known. That's what the Genome Project and the research since then has provided. Millions of variants, some causing disease, some not, are now in public databases. So creating these data did not require any creative patentable process. So all the discussions going on now about reform of Patent Code 101, it's very important to keep in mind that in terms of impact on precision medicine and impact for diagnostics in particular, be very, very careful about doing anything that would overturn AMP v. Myriad decision. Summary: genomic medicine seeks to know an individual's genomic makeup in order to deliver the most effective test or treatment to the right patient. It's essential to precise and individualized diagnosis, therapy, and prevention. And diagnostics and therapeutics are both avid consumers of the research product that is coming out of the efforts of the NHGRI and many other funded operations that are generating the raw material that are being used to create a whole new world of medical care. So thank you. I'm going to turn this over now to my colleague, Simon Tripp.
Thank you very much, Bob, and thank you to all of you today for the opportunity to present our findings. Genomics specializes in performing detailed examinations of science, technology, and associated advanced industries that have a substantial impact on society and our national economic growth. And it's certainly always a pleasure to be able to communicate the impacts of key science and technology fields where the United States sustains a powerful leadership position. As has been said, it's now more than 20 years since the publishing of the draft Human Genome sequence. My colleague, Marty Grieber, and I profiled the impacts of the Human Genome Project in a 2011 study, and it's certainly been a pleasure to be able to examine the tremendous progress that has occurred since then. In the past decade, we've seen dramatic growth in the scope and applications of human genetics. And today we're able to quantify the growth in terms of the impact being generated within the US economy, and also profile the wide-ranging functional applications of genetics and genomics across biomedical sciences and other human application areas. Our report contains two distinct pathways of analysis. One considers on the left the large-scale economic impact of research and industry activity that takes place within the US in human genetics and genomics. Here we use the standard econometric technique of input-output analysis to look at the economic impacts. The other pathway on the right-hand side here describes what the research and industry activity actually is achieving in terms of advancing human health and well-being and other positive impacts. So both of these pathways tell an important story within the report. Looking at the economic impacts first, we drive the economic model, the input-output model, using employment, with our employment numbers collected using a number of different in-depth analyses and multiple data resources, because there is no single industry code for genetics and genomics. We had to build the data from the ground up. We find that the genetics and genomics industry contains over 152,000 direct jobs now. And there's approximately also 14,000 jobs in the academic and federal research space focused on genetics and genomics. Notably, because of the technical and precision nature of the work, those jobs are extremely high paying. They actually have an average compensation level of over $130,000 per job across those 152,000 jobs. This slide shows our, some of the top line economic impact findings from the study. There's, of course, much more detail in the full report, which is now available at the American Society of Human Genetics website. We can see that the overall employment impact in the US economy now stands at 850,000 jobs because of the multiplier effect, the re-spending of the original dollars as they circulate through the economy in the supply chain and other locations within the economy. And that overall economic impact number is now $265 billion, an extremely large industry. Notably, when we compare the current core investment by the federal government in human genetics and genomics research, we can see that the government's actually getting a very positive return on its investment, not just in terms of the growth of the economy, which is the primary impact, but just in terms of a 5-to-1 return in terms of the tax revenues the government gets back from this sector. So investing in genetics and genomics is paying off in so many ways. Now, the economic growth being generated by the sector is certainly very important, but the science and the technology is aimed at achieving real progress in human health. Within the report, we examine the multiple areas in which human genetics and genomics progress is contributing to advancements in health and well-being across society, the kinds of examples that Bob talked about earlier. It turned out to be a rather complex assignment because there's so much happening. We ended up classifying the areas that we could see into 8 primary domains of functional activity, each of which are advancing biomedical science and proving invaluable in opening up new pathways to the improvement of human health and well-being. Those include, circulating around this diagram in a clockwise fashion, mineable big data, where very large data sets of multi-patient data from millions and millions of patients who have now been sequenced provide tremendous insights into those gene variants that Eric and Bob have talked about. It's being used for identifying predisposition to diseases and disorders through carrier screening of prospective parents, in neonatal child and adult screening. It's, of course, being used very effectively in diagnostics. Whoops. In diagnostics for diagnosing diseases and disorders that have genetic signatures. We're really seeing very profound benefits being generated here, especially in terms of providing diagnosis of rare diseases and curtailing the difficult diagnostic odysseys that patients and families previously had to endure. And rare diseases impact a tremendous very large patient population. Between 25 and 30 million Americans have a rare disease, and it's about 350 million people worldwide. Rational drug development is another key area, and that there genetics information is informing really precise molecular targeting in drug design and helping to advance new medicines into clinical practice. Pharmacogenomics, also known as personalized medicine, is enabling the personalized prescription of drugs best suited to our individual genetics and genomics, increasing their effectiveness and reducing negative side effects. Then there's the emerging area of gene editing and gene therapy, helped by the new technologies of CRISPR-Cas9, where genes associated with disease can be modified to effectively cure the disease. It's a very fascinating area. And then two sort of emerging areas in terms of human microbe interactions and the two-way effect occurring on the human genome through those interactions, and also the effect that the environment and the different pathways we each take through life with our experiences have an impact upon our genome and our health. Within the report, we describe the importance of each of those areas. We include examples and case studies of some very high impact applications. What we ultimately find is that human genetics and genomics has very much reached an inflection point. It's at a point now where advancements in the field are leading to widespread clinical application and the improvement of the practice of modern healthcare. We would also note that human genetics and genomics do not just find application in biomedical domains. We're also seeing human genetics and genomics being applied in other areas, such as forensic science, anthropology, and evolutionary biology, and in paternity testing as well. I would note that genetics and genomics as a field obviously extends beyond the human, as our report emphasizes human genetics and genomics exclusively, but of course it's also a tremendously important area of science and technology in agriculture and veterinary sciences, for industrial biotech and environmental biotech as well. I think it's interesting to note that recent poll results reported by Research America and by the ASHG show that the public really gets it in terms of the power and promise of genetics research to deliver positive benefits for society and for individuals. The plurality of respondents, as you can see on this chart, are curious, they're hopeful, amazed, optimistic when thinking about the field and its future. So our findings in terms of the economic and functional impacts certainly show the public has a right to be optimistic. Human genetics and genomics as disciplines have had and will continue to have really profound impact in terms of advancing biomedical research and improving the clinical practice of medicine. Millions of patients are benefiting from the advancements already made, and new developments promise much more. Notably, while doing good for humanity, these fields are also doing good for our economy. Over 850,000 jobs are supported, over $265 billion in economic impact generated. We can see that in just 10 years, the impact has expanded fivefold over what we saw when we did our original study on the Human Genome Project in 2011. We do not anticipate that this growth will slow. The science is opening up, as Eric said, just sort of the tip of the iceberg in many respects. And there's just so many interesting areas of inquiry and applications across healthcare that in many respects, we're just getting started. It's certainly hard to do justice to a nearly 100-page report in a short presentation like this. And I do hope that many of you will choose to access the full report to learn more about this really exciting and dynamic US economic sector. Thank you, and I'll turn over to Gail for more conversation.
I'd like to thank all the speakers so much. As a physician who takes care of patients with both common and rare diseases, I see so much how genomics is changing the way we take care of patients, and I'm glad we had an opportunity to share that with others today. We're going to move into the panel discussion section of our presentation. So if you have questions, please submit them in the question and answer panel that you have at the bottom of your screen, and we'll take questions while we have time left. The first question I'll direct to Eric. It says, how has human genetics helped with addressing the COVID pandemic?
Yeah, no, I appreciate the question. And I could say that throughout the pandemic, I've repeatedly been so delighted to see how threads of genomics have influenced many aspects of our response. I'd even start with maybe one that will catch people by surprise. It's not even a scientific one. It's a cultural, a scientific cultural one, in that genomics promoted so aggressively data sharing. And I think one of the signature key aspects of our COVID response was getting that first sequence of the coronavirus out quickly to scientists around the world who could start figuring out many things, including the design of vaccines. And so that, I think, sort of, it was one example. Of course, the diagnostic aspects of coronavirus and COVID-19 have involved sequencing technologies along the lines of the technologies I spoke of that have really come to the fore, have been adapted for sequencing a very teeny viral genome and done on a large scale. And then many aspects of the fact that the major workhorse vaccines, are RNA vaccines, are based on lots of tools of genomics, both in their creation and their scientific study. So I mean, those are some immediate examples of where genomics has played a role. And there's some longer term studies that are now ongoing to try to see if we could understand why people respond so differently to this virus, and whether any of that is encoded in spelling differences in individual genomes.
Yeah, and in fact, I'm doing some of that work myself, funded by NHGRI.
There you go.
Hopefully we'll learn something. The next question I have, I think I'll direct toward Simon, which is, what do you think have been some of the biggest factors contributing to the growth of genetics and genomics since the completion of the Genome Project?
Yeah, I think the speed of genome sequencing, as the slide that we showed earlier, where it's even exceeded Moore's law in computer processing speed, has really contributed. Being able to make whole genome sequencing affordable just makes the science more affordable, and then it enables its movement into clinical practice. So for me, that has been the signature, the technological capability. And this is an industry in which the US is a leader. This is an area where the federal government invested to do the science originally. Industry then leveraged that basic research to fundamentally advance an entire industry. And today we have companies like Thermo Fisher and Illumina and Pacific Biosciences and others that are really the worldwide leaders in this, generating these economic impacts that we've talked about. It's a tremendous success story.
Thank you. Bob, do you have anything to add to that?
Well, I think the other things that have really contributed have been the culture of data sharing that Dr. Green talked about. I think that's been a really important contribution. And the other is the willingness of the American people and other people outside the United States to participate in the research projects, clinical research projects, which allow us to determine what the value is, what the utility is of genetic studies, and show that it actually benefits outcomes and improves health. So there's been a lot of contributions. The technology that Simon talked about was absolutely necessary to make this happen. I mean, in my laboratory, we can sequence hundreds to thousands of genes in a few days, compared to what the situation was like 10 years ago, or not to mention 20 years ago.
I also think
the parallel, and I think, has been the fact that data sciences has advanced so rapidly. I mean, now we have artificial intelligence, machine learning based approaches, because the amount of data generated through sequencing is so massive that you have to have these advanced analytical tools developing in parallel to be able to make sense of the data that's being generated.
Yeah, and I think the other aspect, Simon showed a slide talking about how the public has gotten very excited about genomics. I think it's some work coming out of ASHG and Research America. And some of it, I think, is along the lines of what we've been talking about, the potential of medical applications. I wouldn't completely discount some of the fun aspects that have come out of genomic discoveries and the cheaper technologies. For example, ancestry. I talked to lots of people, neighbors and not scientists. They've gotten very interested in ready access to ancestry information about their family through these ancestry companies, or even popular press coverage of stories about, Neanderthal DNA that all of us carry in our, in our genomes, and sort of the curiosities of unraveling aspects of our ancient our ancient ancestors as well as our more recent ones. So those fun ones, which are not health related, I think do spark interest, especially I think in younger folks who find this very intriguing.
Thank you for that. Eric, I wanted to ask you, in NHGRI's new strategic vision plan, you lay out some bold predictions for 2030. You want to share those?
Well, I'll certainly share them. I'll put in a selfless plug of people. We put out 10 bold predictions of what might happen in the next 10 years, which included deliberately things that were really, I would describe them as beyond stretch goals, because some of them were so audacious. And we put those out, not so much because we think they're all going to come true. In fact, I'm sure very few of them will truly come true, but it really represents things we're lunging for, things we are reaching for. Among them were topics we talked about today. I mean, one of the bold predictions talks about how, getting a genome sequence will feel just as routine as getting a CBC today. That's not such a crazy idea 10 years from now, that a patient would get a genome sequence ordered as readily as they get a CBC. We also talk about, other aspects of genomics, which we didn't really speak as much about today, especially around health equity. Can we imagine 10 years from now where everybody's benefiting equally from genomic advances, that we are able to reduce disparities, or at least not exacerbate disparities that are all too common in our medical system today? So I really encourage people to take a look at these 10 bold predictions. I'll also put in a selfless plug. We've gotten so much attention to those 10 bold predictions. We're actually in the middle of a 10-part seminar series. So if you go to our website and Google, you can see we're videotaping all of them. We've had about 3 or 4 of them so far. They're going to go on for the rest of this year on about a monthly basis. And each one of these 10 seminars involves 2 speakers who speak to one of the 10 bold predictions. And so it's really been fun to unpack it a little bit. And I really think it has really sparked people's imagination in a number of different areas, some social, some scientific, some medical, some technical, just a full range of things that we can anticipate in the next decade.
Eric, are there things that we are not doing now that we need to be doing to realize those predictions?
Well, that's the big question, which we are really left thinking about. So, certainly the strategic planning process that we did, sort of in many ways, it gives you answers to a lot of the things you need to get to. In some cases, it's not entirely clear how to get there. I mean, some of the issues, Simon mentioned some of the exciting areas coming up in machine learning and artificial intelligence and just data science in general. It really speaks to actually one of the slides I showed. We still have, and Gail, you're in the middle of all this, as is Bob, is we still have this gap between the technical ability to generate that genome sequence on a patient and being able to just as quickly operationalize an interpretation of it. And that is as much a data science challenge, but it also raises a bunch of challenges related to, literacy of the healthcare professionals, literacy of the general public around all these aspects. So, there's a lot of steps we need to be working on. Some of them are very technical, some of them are very social, and we also are constantly dealing with the complexities of infusing this into healthcare systems, which are a complicated set of ecosystems. So we're sort of swashbuckling our way through. But I think one of the main messages I would give you, and it's very much embodied by our new strategic vision, it's not all about research projects. It's not all about just taking money and putting it into research. There's a lot of other things that need to be done. Bob even touched on some of it related to some legal aspects of patents. So many different things have to be done outside of the research projects to see all of this come to reality.
Yeah, and I might add the genomic literacy of primary care physicians to the genomic literacy of patients, because we have some work to do there.
Yep, all healthcare professionals.
So I want to ask Bob, what cutting edge medical advances do you see coming in the next several years that use genomics in healthcare?
I think the primary thing that is happening already and is going to be happening on an accelerating basis is people getting genetic information at a time when they may not necessarily have a particular disorder or a particular disease that they're concerned about, but to have that information and therefore allow it to inform their healthcare going forward. Pharmacogenetics, genomics, for example, that Simon talked about, is a clear example. If your doctor is going to prescribe a drug for you, that's not the time when he should be testing whether you have a genetic alteration that's going to change whether that drug works for you or causes an adverse event. That's something you should already have. You should already have it in your electronic medical record, readily accessible, easily used, with clinical decision support. So I would say the biggest change that's going to happen is genetics and genomics is going to change from the last thing you do in making a diagnosis to the first thing you do to protect people's health.
Bob, do you want to say anything about or Gal want to say anything about the challenges of integrating all that into electronic health records when our electronic health record systems in the US and everywhere in the world are pretty complicated and not so easy to manipulate?
Give Bob the first shot.
Okay. Well, I mean, even when a single company sort of dominates a good, substantial fraction of all the electronic medical records in this country, each instance, each installation is actually different, one from the other. And I have to say that those companies don't necessarily make it easy for companies like mine or other companies to be able to speak with the electronic medical record very easily. I should also say that individual hospital systems are very concerned about the security of their data. And so there are very powerful information technology barriers that are put up. And so for example, it's a one-on-one process for us to have our systems brought into an electronic medical record in any one institution, and often takes weeks, sometimes even months, to negotiate around that. We need some sort of universal, secure system to be able to do that simply and easily, and we're not there yet.
So I would add that one of the real promises right now is using genomic information in prevention, as Bob mentioned, preventing bad drug reactions, but also preventing diseases by knowing who needs more screening, who needs screening earlier. And that needs integration into the medical record. So an ounce of prevention is still worth a pound of cure. It is still much cheaper than gene therapy to prevent illnesses. So we need to get around the technology things that are blocking that. We just have a couple minutes left. I want to make sure if any of the speakers have something to add. So Simon, a last thought from you.
No, thank you very much to the American Society for Human Genomics and the other sponsors of the research for bringing us in on this project. It's always a pleasure to be able to talk about an area of science and technology where the US is leading and has the ability to continue to lead into the future.
Thank you. Eric?
Oh, thanks, Gal. I just want to go back to our bold predictions because I'm smiling here when we were starting to talk about the challenges of getting access to your own genomic information through your electronic health records. Going back to the bold predictions, our bold prediction number 8 in our strategic vision was an individual's complete genome sequence along with informative annotations will, if desired, be securely and readily accessible on their smartphone. There's an aspirational goal for the next decade. That's why we put it as a bold prediction.
Fantastic. Bob, last words?
Yeah, this is not such a bold prediction. I just think it's something that is going to happen, is happening now. And that is that genetics and genomics will be totally integrated into healthcare from a strongly preventive point of view. And that all of medicine is going to be using genetics and genomics for the work that they do.
That's a great word to end on that I'll endorse. I really appreciate all the speakers so much on the behalf of the Society. I really want to thank everyone for their participation, the audience for their attendance, and I would ask anyone in the audience to contact ASHG if you have further questions. And we are always a resource for you, not just this week, but always. So please look to us if you need information about this field. Thank you again, everyone. Bye-bye.
Bye, everyone.